Okay, so welcome to lab one of the course G08 on metagenomics applied to viruses. What we're going to focus on here is some background on microbes and metagenomes, just to remind you about aspects of that and metagenome sequencing. And then we're going to move to the main aspect of the practical of metagenome taxonomic classification using Kraken 2, including getting our data, subsampling our data so that we're not dealing with a huge size, and then understanding how Kraken 2 can work and how we can extract information from Kraken 2. So if we look at the broader aspects first, we know that metagenomes are very interesting in terms of their try to discover new genes and new variants that exist in different environments, whether they're livestock, clinical, environmental, or beyond. And we know that microbiomes refer to all the microbes in a particular niche, whereas a virome refers to the virus particles in a particular niche. And distinct from that, we have the metagenome refers to the DNA of all the microbes in the niche. So therefore, we can talk about viral metagenomes here is what we're interested in and what we're focusing on. And there are lots and lots of examples of microbiomes that are interesting. They're in us and on us and further afield. And one technical note is that we're not doing viromics as such here because we're not looking at the particles, we're looking at the DNA or RNA that the particles have. So please see the intro videos for more detail on this. But we know that we want to interpret using bioinformatic methods these viral metagenomes that are from some sort of particular animal often. And we want to understand their community structure and function. And we want to make this fast, efficient, and effective in terms of understanding their genetic diversity that can tell us more. And where possible, and where metagenomic strength is that it's an untargeted approach that can reduce the biases we incur. So we have this initial setup that we use some sort of source from which the virus came. We prepare our sample, which may contain particular experimental steps to isolate the viral RNA and DNA that we can then extract, sequence, and then analyze. And that those analysis steps can be quite broad depending on what our research question is and what our data is actually able to tell us. One key note is that bacterial metagenomics and virus metagenomics is quite different. So although there is a lot of diversity in bacterial genomes, they are much less diverse than viral genomes. The databases associated with bacterial genomes are more structured and more clear, whereas for virus genomes, it's much more diverse in terms of the areas in which they're kept in and the sources from which they are derived. Marker gene-based studies such as the 16S ribosomal RNA are useful and effective for bacteria, but for viruses, there are no universal markers with the exception of the RDRP gene, which is present in all or most RNA viruses. Our knowledge of bacterial genome structure is much, much better than viruses that can be quite diverse and especially ones that are very rare. The bacterial taxonomy is much more stable and structured than the viral genomes that evolve more quickly. And we know that many virus genomes are more fragmented, whereas bacterial genomes may be complex, but certainly are better understood. Fortunately, for a lot of bioinformatic methods like weed classification, things like Kraken 2 can be effective for that, which is also useful for differentiating viral DNA from bacterial and other contaminants, and also the host DNA as well. For the most part, we're focusing on whole shotgun metagenomics, which can include things like proviruses, host DNA, other contaminants. There are approaches based on virus-like particles, but these are prone to contamination. So in principle, they may give more precise information on the metagenome that's present, for common ones at least, but there may be biases built within there as well. And key aspects that we want to understand better in our metagenomic analysis is to understand what viral specimens are present and what are their functions. And there's different ways of approaching that we'll explore during our practical, including de novo assembly in particular, remapping we've covered elsewhere already, And we'll look at species taxonomics and assignment, searching functional database, and comparing the relative rates of abundances. We know that sequencing makes a big difference, and there are very good methods around targeted amplicon sequencing and probe enrichment methods that are very effective for sequencing particular viral specimens that are of interest or regions of them. And that's different from metagenomics. Metagenomics, there's minimal preparation steps where possible before sequencing, because that incurs biasing effects. Metatranscriptomics is another parallel and complementary technique where it's looking at extracting RNA versus cDNA for sequencing. And again, you're trying to reduce the biases there. Protein-based metaproteomics is also another method based on mass spec and extracting the protein for digestion before mass spec to tell us more about the amino acid structures of the different peptides that are present. And we also have metabolomics, which looks at metabolites, and there's different approaches for that using mass spec or NMR. As mentioned in the introduction, we can take a particular sample and try to explore, after extracting its DNA and sequencing it, to see what taxonomic classification it may occupy. We can then do QC and then do things like a co-assembly, that's of the whole metagenomic sample, or targeted assembly of particular ones that are of interest. And that might use reference information, such as our approach that we're going to apply with Kraken 2. And therefore that uses mapping reads to particular either the same conjugates or particular reference genomes that are of interest. And then there are a lot of things that we can do following that to follow on from there. We know that metagenomics is very, very useful. We saw it in action for SARS-CoV-2. This was a novel virus. There was some related to other viruses, and that average sequence divergence was pretty consistent across the whole genome. And using assembly, phylogenetic methods, and study of its genome structure and genes told us a huge amount more about it. So now we can move on to looking at our approaches for the two labs. So we have one approach that we're focusing on initially here. We can do QC, which we covered in labs before, but is important if we're looking at any new particular sample. We'll do focus on here on read assignment with Kraken 2 and extraction with the Kraken 2 Python scripts to assign context as viral. In lab 2, we'll look at de novo assembly methods and checking for contamination with check-v. So that's approach A, which pivots on Kraken 2 and then de novo assembly and subsequent analysis. And that's different from approach B, where we're going to do de novo assembly of the metagenome first. We're going to use locum search to search these contexts and get more information about them. And we're also going to use check-v to get more information about the context we generated. And then we can compare and contrast the different methods that we used in A and B. So we're trying to taxonomically assign the reads, and Kraken 2 operates on the reads. It is dependent on the database. Unfortunately, on the OROX, we have those databases available. We, where possible, trying to assign the reads at the level of genus or species, and Kraken 2 will assign the lowest common ancestor. So in a sense, Kraken 2 is a supervised method that assigns reads to species based on k-mers if they're well known. If we have something that's extremely unusual and not present in the Kraken database, that is where it will be limited. This is different from metagenome bidding. So that in that approach, we have in lab part B, we're doing our sequencing, getting our reads, doing our assembly, and then bidding them into different species groups based on the similarity and read depths that we have present. And these bins are all metagenome associated genomes. So that's different from what we're doing. We're using Kraken 2 to tease things apart first. So what we can do after that then is, blast the context, you look up different databases, protein level alignment may be better. And Kraken 2 is certainly effective if you have known viral genomes, but if it's something unusual, it may be arguably less effective there. There's lots of existing databases such as the ENA, IMGM, the NCBI with lots of information there. So your choice of database and reference information really depends on what you're trying to information you're trying to explore. So what's great about Hack Kraken 2 is that it already exists on the Aurochs, which is great, and it uses minimizers of some length L, where L is smaller than K, and K is a sequence length reflecting the set of k-mers from which the minimizer comes from as a representative. So we look at each minimizer against this query sequence, the read, and assign it to the most general taxonomic category that's possible. So that prioritizes genus ahead of species, family ahead of genus, and so on. And the reads could have no hits, they're unclassified, or multiple hits. The more hits they have, the more strength we can have that they're properly classified. And we were able to store these together by linking the minimizer matches and the lowest common ancestors in a single hash table. It does have our error rate because this is looking at fragments of the reads, not the whole reads, for computational efficiency, because this is a complex process. So if we have a k-mer spanning this whole read, we can take the pink region, which is the length L equals 31, and take that in minimizer and look up our k-mers. So that's going to use that k-mer to see what matches are present in the database using hash function and create a table based on that information. So it's going to try to efficiently identify the matches and store each match with its LCA to identify the taxonomic groups to which it may belong. So that kind of gives us information then read by read level of the taxa to which it most likely belongs. And per possible, this will move if there's one shared across the genus, across the species, it'll allocate to the genus. If they're shared across the genera, it'll be allocated to the family and order and so on. So that's its purpose. So our data is real data from a 2017 study in dolphins, whose data I borrowed, where the fecal samples were prepared for viral metagenomics. So there's a question over whether there's any bacteria present. And we know that the dolphins had some nasty gastroenteritis virus that was affecting them. So what we're going to do on the server shortly is use curl to download the original sequences. That may take a little bit of time to download. And then we'll use the reformat s.sh script from bbmap to subsample the reads just to take away 100,000 of them all. You can change that if you want, but if you use the whole data set, everything will take a long time and you'll be waiting. You might need to let it run overnight for Kraken 2. And then what you can do as plan B if you're struggling is just copy the subsampled samples from the metagenomics folder already, and that will save you time. So it's up to you what approach you want to take. What we will do is check firstly that you have access to Kraken 2. So Kraken 2 is very memory intensive. Hopefully you don't get issues during the lab. It uses a lot of RAM and you'll have to run it on the head node because it doesn't work on the compute node. And what you need to do before you run it is make sure you have made the Kraken files and Kraken report directories to which your output will be recorded. So here we have our commands. Our command structure is to have Kraken 2. Our database is in home share. We're just using one thread. We have paired samples where this is the first FASTQ file. This is another FASTQ file. Our output is sent to this folder for the Kraken file. And then the report containing the taxonomic classification is sent to the Kraken report folder. And you see that the names of those files are both sample. So it's just for simplicity to refer to the same sample consistently. So you'll need to change these output names if you're using this for something else. And what we expect to find is about 99.95% of reads are classified, which is unusually high in this artificial setting. So what we'll do is look at that now on the Aurochs server. Okay, so we're going to try those two curl commands to download the FASTQs, and you should see this when they're downloading. It should be relatively quick without any issues. And what we can do is paste in that reformat.sh command, which should be fully accessible for all of you. And you'll see that I already have the samples there, so I'm just going to remove them. So if you are re-downloading them, you may get that, or re-subsampling, you may get that issue. And let it run through, and it's very, very quick. And we have subsampled our reads so that we don't take forever in running all these things. We can loop through the FastQC steps, but we're going to skip that. And what we're going to do is use our command earlier to firstly check is Kraken 2 available for us, and it should be in user bin. And then we can, oh dear. Just use that command to run Kraken 2 that we had before. What you'll see is that it'll say that firstly it's loading information, and this may take some time. So this is therefore a good time for us to think about what output we're going to get from that, and look at that further. So we'll note that this takes some time, and then look further at what output we get from the actual Kraken report itself. So what we can see from the output is that we have reads that may be classified or unclassified. The reads have a header associated with them and will have no taxonomic unit taxonomy ID for the ones that are unclassified, but for ones that are classified, we will find that there's a taxonomic identifier associated with that. Some which may be very long if they're a species, or shorter if they are at a lower level of the taxonomy. And those names will be associated with a number of hits that are present, such as 5 here. Or if we look at this one down here for this taxonomic ID, we can see there that we have 4 hits for this one. So that'll be the kind of output of that we get. And it's worth looking through your file to see what you get for your different matches, and that'll help you solve this puzzle of what potential viral agent is affecting these dolphins. So when you look at that, you'll see this. There'll be a percentage ID here, and it all will start out very high, and it gets smaller and smaller as it gets more resolved. For example, here we're going through bacteria, Proteobacteria, alpha Proteobacteria, Rhodobacteriales, Rhodobacteriaceae, and so on down to a more precise one. So that's what we'll look at shortly. For the next step, what we'll do is take our taxon of interest. What taxon is this? You'll have to look at your Kraken files for that. And we're assigning that in bash, and just check with echo to check that this is correct. We'll make a directory for this as well. That's important. And what we'll do is use the script extract Python Kraken reads, which is in that folder. So you need to copy it over. And it'll have input of the Kraken files. It'll also take as input the this is just for the first FASTQ file. It'll have output. It'll send all those viral reads to this output location. So we're putting in Kraken valid files, sample1 virus.fasq. The -t parameter is the taxon. We're creating out FASTQ output, and the report, the Kraken report is at this place, okay, as well. And we're including the child nodes as well to identify all reads that could be associated with that taxon, because many read and K-mer and minimizer matches will be conserved elements that are present in many different species, genera, and families, and so on. So we'll do that, and we'll do the same for FASTQ too. So you just change the input FASTQ, the output name, and that will mean that our final reads will be in these files. So that's how we can take that information and extract our reads from that. So let's look at that now on the server and our output from Kraken 2 originally. But when we look at our Kraken output, you should see something like this. So we looked at 254,000 sequences, and the vast majority of them were classified. So then if we look at the output, for example, in our Kraken files, you'll see something like this. So it's the classified, that's the read identifier. Next you have the taxon. I mentioned this taxon ID already. and you see it as 13 matches. If we look further down, some matches that are classified with other taxa, um, and so on, we can kind of see through the report that there's different ones that are classified. And then what we can do is look at a Kraken report. And here you see that, move my icon, that the 99.7% of virus, we have a little bit of other viruses that are present. What you see is that 99.6% of them are associated with bottlenose dolphin adenovirus 1, which corresponds to that taxon ID. And you can see the kind of lower the taxonomic context of that particular virus. You see there's a few other things in there, gamma herpes virus, but this is a very, very low level of reads in contrast to the adenovirus above. So maybe present and maybe contaminant is a bit at this ambiguous level. And you can see after here, once we go down to read match level 1, this is just definitely noise. So that's our Kraken report and output. And what we can do next then is create our taxon and assign that. So we have our taxon there and let it run to extract that information for those FASTQ files. You can see this is quite quick because we subsampled the reads rather than doing them all. So that's the first one done. And we can do that for the second one as well. And that's quite quick. If we took the whole file, it would take much, much longer. So bear that in mind. So that's our initial approach for looking at the Kraken data. One thing that we could do then is, for example, we could assign the taxon to be something else, like this one, or what's the one for the herpes virus? For example, that, and rerun these again, but we would have to change the output file to a different ending because otherwise it would allocate those reads to a different output. So that is using $taxon as a taxon. And if we change this for the second FASTQ file as well, that will also extract reads for this gamma herpes virus. And when we look at the numbers of reads, we go into that folder, you see there that we have the file sizes are very, very different, indicating that we have a lot more of this adenovirus that is present than the herpes virus. So we could analyze this herpes virus individually itself using de novo assembly as one to see what's there if that was of interest to us. So it's just in this case, we happen to have the vast majority happens to be a single virus in this particular instance. So that concludes this part of the lab. Please continue with lab 2 to look at using spades to de novo assemble these and the whole metagenome itself.